Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about microscopy part 2, that is fluorescence microscopy and electron microscopy. So in the previous lecture we have already discussed about the introduction part of the microscopy, that what is microscopy, what is the history of microscopy, and we have discussed the two type of microscopy, that is a phase contrast microscopy and a compound microscopy. We have seen the working principle, we have seen the advantages and disadvantages of both of the microscopy. So same in this lecture we will going to cover fluorescence microscopy and electron microscopy. We will see the working principle, we will going to study about the advantages and disadvantages. We will also discuss about what are their applications in different fields of industries. So let's get started with the topic and let's see what is a fluorescence microscopy. A fluorescence microscopy, so since its name suggests, it uses a fluorescence and a phosphofluorescence to examine the structural organization, spatial distribution of sample. It is particularly used to study samples that are complex and cannot be examined under the conventional transmitted light microscope. Fluorescence microscopy image helps to study substances present in low concentration where a high sensitivity is crucial to detect them. So basically it is a type of a microscopy that uses the fluorescence, and with the help of these fluorescence, or you can say the phosphofluorescence, they examine the structure of the different types of samples. So where the light microscope cannot detect or cannot examine the complex or samples, then their fluorescence microscopy is used. So basically you will say in simple language that fluorescence microscopy, it uses the fluorescence, and with the help of these fluorescence it examines the structures that are so complex that they cannot be examined by a light microscope. So the image that this type of fluorescence, this type of microscopy gives is to study the substance that are present in a very low concentration and they have a very high sensitivity to detect them. Now let's see the diagram of the microscope. So this is a type of this fluorescence microscope. So somewhat it looked like this, and they have a detector. Through this detector they detect a sample. They have excitation filter, and they have the emission filter. They have a dichromic mirror, and the object is placed, they have objective lenses, and at the end they have a specimen. So here the light source, it uses a different types of fluorescence, and with the help of this fluorescence this detector detect the sample. Now this is very important, that on which principle this type of fluorescence microscopy work. So the most cellular components are colorless and cannot be clearly distinguished under a microscope. So the basic principle of fluorescence microscopy is to stain the component with dye. Fluorescent dye known as the fluorophorans or fluorochromes are molecules that absorb the excitation light at a given wavelength, usually UV, and after a short delay emit light at a longer wavelength. The delay between the absorption and emission is negligible, generally on the order of the nanoseconds. So the emission light can be filtered through the excitation light to reveal the location of the fluorophores. Fluorescence microscopy, it uses a much higher intensity light to illuminate the sample. This light excites the fluorescence species in the sample, which then emit light of a longer wavelength. The image produced is based on the second light source or the emission wavelength of the fluorescent species rather than from the light originally used to illuminate and excite the sample. So this is very easy that this type of fluorescence generally they uses a dye, or you can say simply you can say they uses a stain component. And this type of dyes that this fluorescent microscope uses, they are known as the fluorescent dye. They are also known as fluorophores or fluorochromes, and basically these two are the molecules. What kind of molecule are they? The molecules that absorb the excitation light, and these excitation light have a particular wavelength. Means at a given wavelength they absorb matlab they work at a given wavelength. Generally they have a UV, and with a very short delay, or you can say with a means after a very short delay, what they do, they emit light at a very longer wavelength. So there is a delay between the absorption and emission that is very negligible, and it's generally of a very nanosecond. Means that type of delay you can easily neglect it. And the emission light, what they do, they can be filtered and through the excitation light, and with the help of this they reveal the location of the fluorophores. This fluorescence microscopy, this type of microscopy, they uses a very much higher intensity of light, and this type of light they uses to illuminate the sample, and this light excite the fluorescence species in the sample, and after excite the fluorescence species of the sample, they generally what they do, they emit the light of a longer wavelength. So the image that is produced, it is based on the second light sources, or you can say on the basis of the emission wavelength of the fluorescent species, rather than from the light that is originally used to illuminate and excite the sample. So this is the working principle of the fluorescence microscope. Now let's quickly discuss how does the fluorescent microscopy work. So if you can see this diagram, you have the stage where the object is placed, you have a dichromic mirror, you have emission filter, you have excitation filter, and you have a light source. So a fluorescent microscopy, it generally work by combining the magnifying properties of the light microscope with the fluorescent emitting properties of the components. So this type of fluorescence microscopy, what they do, they uses a very high intensity light source. What they do, they excite the fluorescent molecule that is known as fluorophores in the sample absorbed. The sample are then what they do, they are labeled with fluorophores, means where they absorb the light high intensity light from the from the source, and what they do, they emit a very lower energy light of a longer wavelength. So the resulting fluorescent light is then separated from the surrounding radiation with filters, allowing the observer to see only the fluorescing material. The resulting image is magnified version of the specimen that is studied. The majority of the fluorescence microscope used in the biology today are fluorescence microscope, Both the excitation and absorption of the fluorescence occur above the sample. Now, what are the fluorescence microscope used for? Means what are their basic application and what are their basic uses? So, a fluorescence microscopy. So they find used in different biology, biomedical, and material sciences. So the unique functionality of the fluorescence microscope, what is that? They help to identify the cells such as microscopic cellular component with the accuracy and details. It is used in the food chemistry to assess the presence, structural organization, and spatial distribution of specific food component in the product. Another use of the fluorescence imaging is fluorescence speckle microscopy. So this is very important. So this type of technology, it uses the fluorescence labeled macromolecular assemblies such as cytoskeletal protein to study movement and turnover rate. Fluorescence microscopy staining also is helpful in the field of mineralogical application. It is routinely used for the study of materials such as coal, graphene, oxide, and more. This is also widely used in the textile industry to analyze fiber dimensions. Fluorescence microscopy is ideal for the study of porosity in ceramic using a fluorescent dye. It is also applicable to study the semiconductor. So as you can see, apart from the used in laboratory, they have also be used in material sciences, biomedical sciences. So basically they are used to study the application of semiconductors. They have a great used in fluorescence speckle microscopy, where they used fluorescence labeled macromolecules such as cytoskeletal protein. And this fluorescence microscopy, it also helpful in the field of mineralogical application, where they are used to study different types of minerals like such as coal, graphene, oxide, and etc. And apart from this, they have great importance in the food industries, where they are helping in the food chemistry, and they are used to assess assess the presence of structural organization, and how the distribution of a specific food component in the product. So this is the uses of the fluorescence microscope. Now let's see the advantages. So the fluorescence microscopy is the most popular method of for staining the dynamic behavior exhibited exhibited in the liver cell imaging. This stem from its ability to isolate the individual protein with a high degree of specificity, or amidst the non fluorescence material. The sensitivity is high enough to detect a few as 50 molecules per cubic micrometer. Different molecules can now be stained with different color, allowing multiple type of the molecule to be tackled simultaneously. This factor combined to give fluorescence microscopy a clear advantage over other optical image techniques for both in vitro and in vivo imaging. So it has a great impact in different types of study, and it is most popular type of microscopy whenever you are studying dynamic behavior exhibited in the live cell imaging. And they have a very much importance where you want to isolate a individual protein, and those protein that have a very high degree of specificity amidst non-fluorescing material. So where and this type of microscopy, it is also used to detect 50 molecule per cubic micrometer. So you can understand this advantages that 50 molecules per cubic micrometer. So there also this type of microscopy is used, and it has a great advantages of using this type of microscopy, where you are have a whenever you want to detect any in vivo or in vitro imaging. So this type of microscopy has a great advantages. Now whenever we are coming having any advantages of any type of a microscopy, then there must be some limitation that is also associated with that particular microscopy. Then the first limitation of the fluorescence microscope is the fluorophores lose their ability to fluorescence as they are eliminated in a process called photobleaching. Photobleaching occurs as the fluorescent molecule accumulate chemical damage from the electron excited during the fluorescence. Cells are susceptible to the phototoxicity, particularly in short wavelength light. Furthermore, the fluorescent molecules have a tendency to generate reactive chemical species when under illumination, which enhance the phototoxic effect. Unlike the transmitted and related light microscopy technique, fluorescence microscopy only allows the absorption of the specific structure which has been labeled for the fluorescence. So these are certain disadvantages, or you can say limitation of the fluorescence microscope. So now before me moving to the electron microscope, let's quickly understand that what all we have covered under the fluorescence microscope. So if we check the one of our slide, so we have covered what is the what is the fluorescence microscopy. We have covered the fluorescence microscopy diagram. We have also studied about the principle and how this fluorescence microscopy generally works with the help of this diagram, and we have also discussed about their uses, about their advantages, and at last we have covered the limitation of fluorescence microscopy. Now let's quickly discuss what is an electron microscopy. So our next type of microscope, or you can say microscopy, this is what is an electron microscopy. This is the most widely type of microscope that is used nowadays in different types of industries. Now let's quickly see and understand what is it. So the electron microscope, before understanding, since its name suggests electron, so here what is it used? Since the in fluorescence microscopy, since the name suggests fluorescence, means we are using a dye. Now here it is an electron, so here we are using a beam of electrons. So the electron microscope uses a beam of electrons and their wave-like characteristic to magnify an object's image, unlike the optical microscope that uses a visible light to magnify the images. So the electron microscopes functions like their optical counterpart, except that they use focused beam of electron instead of photons to image the specimen and gain information as to its structural composition. So here, since its name suggests electron, so here they are using a beam of electrons and their wave like characteristic. So both of them are used to magnify any object's images, and unlike the optical microscope, it generally used to visible it uses the visible light and to magnify the images. And this type of electron microscope, it uses the beam of electron instead of photons to image the specimen and gain information as to its structural and composition. So this is the diagram of electron microscope. If you have seen in different industries, this type of microscope is always there, and it has a great advantages of using this microscope. And this microscope has an electron gun. They have the lens that is known as the magnetic lens. Then they have a electron beam that is generally came from the electron gun. And then they have a sample, and they have one screen to see. So this is the diagram of the electron microscope. Now moving to the working principle of electron microscope. So whenever it's we have seen the working principle, then here also we will going to study about the working principle of the electron microscope. So the working principle of electron microscope, it uses a single arising, or you can say a beam of electrons, and with a sample to obtain information about structure, morphology and composition. The electron gun, it generally what they do, they generate the electrons. The two set the two set of condenser lenses, and condenser lenses focus the electron beam on the specimen and then into the thin light beam. They also helps to move the electron down the column and accelerating the voltage. So generally the voltage is between 100 kV to 1000 kV. The specimen is to be examined is made extremely very thin. Why? At least 200 times thinner than those used in optical microscope. So the ultra thin section of 20 to 100 nm are cut, which is already placed on the specimen holder. The electronic beam passes through the specimen and the electrons are scattered depending on the thickness of or relative index of different parts of the specimen. So here so the specimen that is used, that should be of a very thin very thin, like 200 thinner times thinner than those that is used in the optical microscope. So the denser the region in the specimen, scattered more electron, and therefore they appear darker in the images. Since fewer electrons strike that area of the screen, so in contrast transparent regions are brighter. The electron beams come out the specimen using the objective lenses, which has a high power and form of the intermediate magnified images. The ocular lenses then produce the final further magnified images. Now comes the type of electron microscope. So basically electron microscope is divided they are basically of two types. The first is the transmission electron microscope, that is known as STEM, and second is SEM, that is scanning electron microscope. So in examination or in interviews, these type of questions is always there, means what is the difference between TEM and SEM, or generally they will ask you the working principle of both the type of microscope. So this is very important. Now let's quickly discuss what is STEM. So since the name suggests, STEM, that is a transmission electron microscope. So this type of microscope, it is used to view a thin specimen and through which electron can pass, generating a projection image. The TEM is analogous in many ways to the conventional or you can say compound light microscope. TEM is used among other things to image the interior of the cell, the structure of the protein molecule, organization of molecule in viruses, cytoskeletal filaments, and rearrangement of protein molecule in a cell membrane. So basically this type of microscope is used where the specimen that is used is very thin, so that the electrons can easily pass through them, and and after passing through them they generate the image. So this is somewhat a TEM look likes. So it has an electron source from where the electron will come. Then they have a condenser lenses, they have a condenser aperture, they have a sample, they have one objective lens, they have an objective aperture, selected area aperture, they have a intermediate lenses, they have a projective lens, and at last they have a one fluorescence screen where they can portray the image. Now comes the SEM. So SEM, it is a scanning electron microscope. So they are the conventional scanning electron microscopy, and they generally depends on the secondary electron from the surface of the specimen. So both of its great depth or focus, scanning electron microscope is the analog to a stereo light microscope. So what does they do? They provide a detailed image of the surface of the cell and the whole organisms that are not possible by the TEM. So it can be used for for a particular counting and size determination, and for the process control. It is termed a scanning electron microscope. Why? Because the image is formed by scanning the focused electron beam into the surface of the specimen in the fast raster pattern. So that is the reason they are known as SEM. Now let's quickly see what is the image of scanning microscope. So again, somewhat it is same. They have an electron source, they have a condenser lenses, they have one aperture, they have a scanning coil, they have an objective lens, they have a backscattered electron detector, and at the end they have a sample. And this detector, they are just they have a detector, and they have a scan generator, and they have a amplifier. So that is the image of the scanning electron microscope. Now let's quickly understand the application of electron microscope. So electron microscope are they used to investigate the ultrastructure of the wide range of biological and inorganic specimen, that is including microorganisms, cells, large molecule, biopsy samples, metal and crystals. So they have a very vast applications where they are used to detect the samples of microorganism cells. Sometimes when you have to detect the biopsy samples, so this type of electron microscope is used. Industrially, a electron microscope, they are often used for the quality control and failure analysis. Modern electron microscope, it produces electron micrographs using a very specialized digital cameras and frames grabbers to capture the image. The science of microbiology owes its development to detect the development to the electron microscope. The study of microorganisms like bacteria, viruses, and different types of other pathogens. So there also this type of microscope is used, and they have also used in the treatment of different types of infective diseases. So to detect the type of diseases, this kind of microscope is used. Now let's quickly understand the advantages of electron microscope. So this type of microscope, it has a very high magnification. They are incredible high resolution power. They have a material relay assortment by preparation it is possible to investigate a greater depth of field. They have a diverse application. Now, what is the limitation that is associated with the electron microscope? So the live specimen, they cannot be absorbed, means whenever the specimen is live, so they cannot be observed. As the penetration power of the electron beam, it is very low, so the object should be or ultra thin. For this, specimen is dried out and cut into ultra thin sections before absorption, and because only they can detect the specimen that is very thin, so that is again a limitation. As the EM works in a vacuum, the specimen should be completely dry, means there should be no water. They are very expensive to build and maintain. They require a very high researcher training to operate this type of microscope. So the image that artifacts the resulting from the specimen preparation, so this type of microscope, they are generally very large and cumbersome, extremely sensitive to vibration and external magnetic field. So these are certain you can say limitation of electron microscope. Now that brings to the end of the lecture, and I hope you have understood the lecture. If you have any doubt, any queries related to the lecture, you can simply leave your queries in the comment section below. In the next lecture of the analytical techniques, we will going to discuss all the queries if there is any query. And by the time, follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn. This is our website, that is www.itlsacademy.com. You can simply go onto this website and to get update. And this is our helpline number, that is 7080833450. You can simply call on this number and get updated. Thank you for watching. Please do like, share and subscribe. Thank you. And yes, student, don't forget to press the bell icon to get more update on the lectures that will be upcoming. So yes, don't forget to press the bell icon. Thank you.